This week we'll be looking at both chapters 13 and 14. We'll start with chapter 13, Observing Patterns in Inherited Traits. The beginning of our chapter starts off with another story. This is a sad one about individuals who have a genetic disorder called cystic fibrosis, and it's one of the most common fatal genetic disorders in the United States. And unfortunately, those that have it usually live no more than 30 years. I actually know a colleague whose nephew has cystic fibrosis. The what's called CFTR gene, cystic fibrosis gene, encodes a protein that moves chloride ions out of epithelial cells. So basically it's a membrane protein, a channel protein that moves the chloride ions that are in the cell to the outside. And when it does this, water usually follows, and so that makes the surface of the epithelial cells, which cover many of our organs, nice and wet, so that when mucus is sliding by, it actually does slide on this moisture. However, in people who have a mutation called the deletion F508, that's what the deletion is called, which basically just affects one codon, which should code for a phenylalanine amino acid. It disrupts, usually in people, it disrupts the protein is actually made, but it never reaches its spot in the cell surface. Therefore, it never gets to get to the cell surface and do its job of removing chloride ions and water from the cell. So these people basically don't have that nice wet surface on their epithelial covering cells of their organs. So when mucus goes by, it actually sticks and it builds up. And this can affect organs such as the lungs, the digestive system, the reproductive system, the liver, and the pancreas. And with the lungs, you can really see the problem with needing to keep our tubes in there free so that air can pass through. But with the mucus building up, it can become difficult to breathe. And so people who have this have to go through usually some treatments, antibiotics, chest thumping, posture changes, etc., and a strict regimen of all that in order to ensure much life as they can get. It can also lead to bacterial buildup in these passageways, which makes these people more prone to infection. In this chapter, we'll begin to find some answers to why it is people with one gene for this don't have problems, but people with two genes, one from mom and one from dad, why they do have problems. And we'll be looking at a lot at inheritance in this chapter. First, we'll go back and do a little history. Gregor Mendel is sometimes known as the father of genetics, and he lived in the 1800s. It's kind of interesting with his lifestyle. He actually wanted to be a teacher, was interested in nature and science, but he never succeeded in being a teacher, but he still wanted to study science. So he ended up joining the monastery, which was very common back at that time. People who had joined the monastery, monks and so on and so forth, were usually able to study and were some of the most educated people of the time. So he joined the monastery and actually got to work in the greenhouse area. And this is where he started his infamous pea plant experiments with inheritance. So basically, he in his garden started pea plants that bred true for a particular trait. So, for instance, let's say that he had pea plants that were both um, white and purple with their blossoms. I'm going to pull up a picture of that. Look at it this way here. So this would mean that like, let's just say, for instance, he had to make sure they were pure breeding. So on one area of the greenhouse, he would grow purple flowered pea plants until they only ever produced purple flowers. On another area of the greenhouse, he had white flowered pea plants, and he kept breeding them until they only ever made white flowers. So now he's got two areas. One only ever has purple, the other only ever has white. So it's said then that they're pure breeding, or what we call homozygous for their trait. Homozygous purple and homozygous white. Then he cross-pollinated them, finally. That actually took about 2 years to make sure that they were each indeed homozygous. And then he cross-pollinated, and back then one might expect to then get a mix of the parent traits, such as maybe a light lavender, but instead 100% of the offspring were indeed purple. Now you'll see some genetic terminology here on the side. The P generation means the parent generation. The F1 generation is what's called the first filial generation, F-I-L-I-A-L, or the first generation of offspring. And they were 100% purple. So this might have had Mendel perplexed, because there was a blending hypothesis at the time where children, of course, when you look at them, you can see a blend of the parent traits. So they thought, well, if I mix purple with white, I should get kind of a lavender, but instead they were all purple. Well, Mendel took it a step further, and he took this first filial generation of all purple plants and he crossbred them together. I know that sounds kind of gross because they have the same parents, it kind of looks like here, but that's okay, they're plants. And here's what he got. So this is called the F2 generation, or second filial generation. And he didn't get all purple this time. The white trait actually came back in about a quarter of the flowers. The other 3/4, though, were all purple. And that's a little summary of Mendel's first experiments. And he did this with many other traits in flowers. So again, back to our regular notes. Mendel started with garden pea plants that bred true for a particular trait, meaning the trait stayed the same in all the descendants, generation after generation. We already said that means that they were homozygous. Then when Mendel cross-fertilized the pea plants with the different traits, the purple and the white, that's when he got all of just one color, the purple. And then Mendel concluded that hereditary information is passed from one generation to the next in discrete units. That means either or, one or the other units. Today we know individuals of a species share certain traits because their chromosomes carry the same genes. The DNA sequence of a gene occurs at a specific location on the chromosome that's called a locus. Remember, as diploid organisms, that we have pairs of genes because we have pairs of chromosomes. one from mom and one from dad, with the exception, remember, of the X and the Y chromosome. If you're a female, you still have two copies of the X chromosome, one from mom, one from dad. But if you're male, then remember you have one copy of the X chromosome and one copy of the Y chromosome. Therefore, the genes on the X and the Y do not match up. So any genes that are on the X and the Y chromosomes for males, you only have one copy of each of those genes. But considering the rest of the genes for which we do have a pair of each of those, when they are identical, they're called homozygous. And when you get slightly different versions of the genes, or alleles, slightly different versions of genes are alleles, then we call that situation heterozygous. Often we use letters to symbolize alleles or genes, such as, take for instance the letter A. For being homozygous, we could say you are big A, or little a. And if you're heterozygous, you're one of each, big A and little a, big A or lowercase a. Any pair of genes on homologous chromosomes may vary as alleles. Homologous chromosomes, remember, are the ones that match up, like pair number 1, one from mom, one from dad. Pair number 2, again, one from mom, one from dad, all the way down to the X and the Y chromosomes. And different alleles can result in different versions of a trait. For instance, blood type is a gene and comes in 3 alleles. There's the A allele, the B allele, and the O allele. Then there's some that only come as discrete traits that are either or, such as having freckles or not having freckles, either or, one or the other, having attached earlobes or unattached earlobes. The particular set of alleles that an individual carries is their genotype. So for instance, if you have big A, little a, that's called your genotype. The observable traits that I can see, such as purple flower color, that's your phenotype, or physical traits. Offspring resulting from a cross or a mating, often called a monohybrid cross or a dihybrid cross, between homozygous individuals with different traits are called hybrids. Often expression of one allele influences the expression of the other, and the outcome is visible in the hybrid phenotype. A hybrid, again, is offspring of a cross between two individuals that breed true for different forms of a trait. A heterozygous individual is another name for a hybrid. An allele can be dominant when it masks that of a recessive allele that's paired with it. So for instance, going back to the letters that I've been using, A, that capital A would be the dominant allele, and lowercase a would be the recessive allele. And if you were heterozygous, big A, little a, then you would show the dominant trait, and the little a would be masked. You would be a carrier of that and maybe not even know it. Again, modern genetics started with Gregor Mendel with his work with pea plants. He was very meticulous, and that was good. It gave him clues that heritable traits are specified in units. They didn't know about genes yet. The units, which are distributed into gametes, which are sperm and egg, in predictable patterns, were later identified as genes. When Mendel crossed pea plants that bred true for purple flowers with plants that bred true for white flowers, again, remember, all the offspring had purple flowers. So we would say that the starting pea plants, the purple ones would have been big P, and the white ones would have been little p. They would have been homozygous for their traits. The big P one would have been homozygous dominant. And the little p would have been homozygous recessive, though he didn't know that until he actually made the hybrids and saw that when he mixed the big P's with the little p's, that they were all purple. That these big P, that was purple was the dominant trait. The allele for purple is dominant over white. So again, any of the hybrids or heterozygotes would have had purple flowers. So this led Mendel to his law of segregation. So when homologous chromosomes separate during meiosis, the gene pairs on those chromosomes separate also. So what this means is that when sperm and egg are made, that let's say that your chromosome pair number 1 that you have, both your number 1s will not end up in one egg or one sperm. They have to be split up into separate gametes, because you can only give your children half of your genetic info, not the full amount. Therefore, then each gamete that forms carries only one gene of a pair. When homozygous dominant and homozygous recessive plants are crossed, for instance, big P times little p, there's only one outcome possible. Big P would be the genotype, and the phenotype would be purple in the F1 offspring. Again, let's see that again. Crossing the purple with the white, each homozygous for their trait, creates the purple hybrids that are all heterozygous in their genotype. And then in the F2 generation, the white returns. Remember, in order to be white, you must be homozygous recessive for your trait, little p. Whereas purple can be either big P or big P. A Punnett square is used to predict the genetic and phenotypic outcome of a cross. Breeding experiments use test crosses to determine whether the tested individual is heterozygous or homozygous. For instance, if Mendel wanted to have known with the flowers which one was dominant, then he would have done his experiment like he did, making sure all of the purple ones were true breeding and all of the white ones for true breeding or homozygous for their traits. And then when you cross them, that's the test cross. And by getting all purple in the F1 generation, he could tell that purple must have been the dominant trait. And that's what a test cross is like. Often in breeding programs, that is used. Another breeding experiment, a monohybrid cross, checks the dominant relationship for a single trait. That's basically what I just described. Breeding experiment, again I'll read this, the monohybrid cross is a breeding experiment in which individuals identically heterozygous for one gene are crossed, and the frequency of traits among offspring offers information about the dominance relationship between the two alleles. So let's say we have a monohybrid cross that we're doing, and we have two big P plants. So for instance, if I go back here, so we know that in the F1 generation, these are big P because we crossed big P with little p. So we know all of our F1 generation is big P. So let's say we take two of these plants and cross them. And here's some of the different results that we can get. We have two big P's together, and of course we'll get big P, big P. Big P with little p, of course, makes genotype big P, little p. So in our first one here, these would be purple. Second result, these would be purple. Third result, these would be purple. And lastly, these would be white. So if we were doing a Punnett square, then 1/4 of the offspring, or 25%, would be homozygous dominant. Half would be heterozygous, purple. And 1/4 would be homozygous recessive, or white. And this is what Mendel got in his F2 generation. Also a 3-to-1 ratio of purple to white. 3 to 1. And this becomes the basis of Mendel's law of segregation. Then came Mendel's theory of independent assortment. So when homologous chromosomes line up during meiosis, so again, that's the making of sperm and egg, your homologous chromosomes should separate into separate gametes, separate sperms or separate eggs. Either one of the pair can end up in a particular nucleus. Thus, gene pairs on one chromosome get sorted into gametes independently of gene pairs on other chromosomes. And Punnett squares can be used to predict inheritance patterns of 2 or more genes simultaneously. So basically this is saying that of all 23 pairs of your chromosomes, all of them have to be split, the homologous pairs split into separate gametes. So let's take pair 1 and pair 2 for instance. If we numbered them, pair 1, if we had pair 1 A and B, and then we had pair 2 A and B, who's going to end up on the, again remember in mitosis, the metaphasal plate, when they line up in the middle like that, the homologous pairs in meiosis actually line up together and then get split apart into separate chromosomes. So if 1B ends up on the same side as 2B, they would end up in the same cell together. But you could also get the result that 1A ends up with 2B. I hope I didn't already say that one. I wish I could write these out and then I could see it better. But it has to do with what side they line up. So you've got all 23 pairs, and whether they end up on the left or the right, it mixes things up, and it's all independent assorting. In a dihybrid cross, individuals identically heterozygous for alleles of 2 genes, or dihybrids, are crossed and the traits of the offspring are observed. So this is looking at 2 genes at one time instead of just one. It gets a little more complex. That's why it's called a dihybrid cross. Di means 2. But sometimes breeders want to know what traits offspring are going to end up with. So let's start with one parent plant that breeds true for purple flowers, and let's team it up with another trait for stem length. So purple flowers with tall stems, and they're true breeding for both their traits. then one that breeds true for white flowers and short stems. Each plant makes only one type of gamete. So remember, you can only pass on half of your genetic information. So a parent cannot give both big P's to their offspring. They can only give one. And same with the T's, only one of the T's. So this parent is making gametes, egg or sperm, with only big P's and big T's in it, while this parent is only making gametes that have little p's and little t's in it. So we say their gametes are either this one, big P, big T, or little p, little t. So when we mix that together during sexual reproduction, big P, little p is our new combination. So what color will that be? If you answered purple, you're correct. And now our new combination is big T, little t, and what stem length will that be? And if you answered tall, you are correct. They'll have purple flowers and tall stems. So again, this is just looking at 2 traits at a time. You can also have trihybrid crosses and look at 3 traits at a time, and tetrahybrid crosses looking at 4 at a time, but that gets quite complex. So we'll stick with monohybrid and dihybrid crosses for this chapter. Now, just like we did in the experiment before, we'll cross 2 of the F1s. So going back to this slide, these are our F1s, big P, big T, little t. We'll cross them. They're plants. Remember, it's okay to do that. And now we get this ratio, 9 to 3 to 3 to 1. 9 will be tall with purple flowers. 3 will be short with purple flowers. 3 will be tall with white flowers. And 1 short with white flowers. And then Mendel discovered that 9 to 3 to 3 1 ratio in his dihybrid experiments and noted that each trait still kept its individual 3 to 1 ratio. So the purples were still always 3 to 1 and the tall always 3 to 1. Each trait or gene pair sorted into gametes independently of each other. So basically, the tall and the color did not affect each other. That's his law of independent assortment, meaning during meiosis, that's gamete making, members of a pair of genes on homologous chromosomes get distributed into gametes independently of the other gene pairs. Again, the gene for purple has no effect on the gene for white. Or excuse me, the gene for tallness. Here's an example of a dihybrid cross. This is the wrong way here, so we would say this is actually the way it works. That this is actually a trait for yellow and green seeds and round and wrinkled seeds, where yellow is dominant over green and round is dominant over wrinkled. And so our F1s all have the big Y, big R, little r. And when gametes are made by those individuals, they can come in these different combinations. Big Y, big R, big Y, little r, little y, big R, and little y, little r. And you can find these gametes by using the FOIL method that you may have learned in math, maybe years ago for me at least. Remember the FOIL method? First, outside, inside, last. So first is big Y, big R. Outside, big Y, little r. Inside, little y, big R. And last, little y, little r. So these are all the possible gamete possibilities from our heterozygote here for the dihybrid cross. And so we're going to mate two of those individuals. So here they are. And now we write all of this individual's gametes across the top and this individual's gamete possibilities across the side. And each of them, because they have the same genotype, will have the same types of gametes. And so let's look at what their babies might look like. And so we fill them in. They might be that one or that one. Look at all the possibilities here of genotypes for their offspring, for yellow or green seeds or round and wrinkled seeds. Alright, so there's our dihybrid cross. Punnett square all nice and filled in. And so these are all going to be the nice yellow round seeds there. And you can see where the yellow wrinkled ones showed up. And the green round and the yellow wrinkled. And then the green wrinkled, which is the completely homozygous recessive trait. So if you count them up for their phenotypic ratios, that's their physical characteristics, there should be 9 that are yellow round, 3 that are yellow wrinkled, 3 that are green round, and just 1 who's green wrinkled. So there's our 9 to 3 to 3 to 1 phenotypic ratio with the dihybrid cross, showing us again that these genes, at least the genes chosen here with these pea plants, don't affect each other. They assort independently into gametes. Mendel was actually lucky because he chose traits on pea plants that are actually genes that are far apart on chromosomes. And when genes are far apart on chromosomes, they do tend to assort independently. It talks about crossing over here when gametes are being made, and let's say at the metaphasal plate, your two number ones line up together, the one from mom and the one from dad. Bits and pieces of each of those chromosomes actually tend to swap places or swap chromosomes. That's called crossing over, and it helps to mix up mom and dad's genes that you got even more so as you put them into sperm or egg. However, genes that are really close together kind of get chunked together and then tend to cross over and swap onto the opposite chromosome. And so we say when that happens that they are linked. And those do not assort independently, and we call those linked genes. And so they tend to be inherited together. Luckily, though, Mendel actually chose genes that are far apart on chromosomes and are not linked. Otherwise, he would have got some totally different results, which might have thrown him off and maybe halted our knowledge of genetics to a later date. But pea plants were a good choice. From dihybrid crosses again, remember pairs of genes on different chromosomes are typically distributed into gametes independently of how other gene pairs are distributed. Breeding experiments with alternative forms of two unrelated traits can be used as evidence of such independent assortment. Beyond simple dominance, well, sometimes the old rules don't fit and you have to change them. Sometimes there's exceptions to the rule. And basically, Mendel chose pea plants, which were a good choice, and he chose genes that were discrete, that were either or, and displayed what are called simple dominance or complete dominance. So purple is completely dominant over white. It masks the white allele. or tallness is dominant over shortness. Tallness masks the short allele, the recessive allele. But there's other patterns that exist that we have found that are not so simple, and we'll talk about each of these right here. For instance, there's codominant alleles, meaning that neither allele is completely dominant over the other. For instance, in the ABO blood typing group, A and B are actually dominant together. And this is why some people have type AB blood. The A allele is expressed as well as the B allele. And so their blood cells show both phenotypes of the A and the B allele. However, both A and B are completely dominant over the O. O is actually recessive to both of those. So somebody who has the combination of the A allele and the O allele will actually be type A blood. Somebody who has the combination of the B allele and the O allele will be type B blood. The only way you can be type O blood is if you are recessive and you have both O alleles. For instance, I have type O blood because my mother gave me an O allele and my father gave me an O allele. This is a multiple allele system as well, besides being codominant with the A and the B, and both of those completely dominant over the O. So a lot of situations going on there with the blood types. In incomplete dominance, the heterozygous phenotype is between the two homozygous phenotypes. So this is very similar to the codominance. So remember, in codominance, with the A and the B alleles with blood type, both the A and the B alleles are expressed. So on red blood cells of people with type AB blood, they have what's called a glycoprotein, carbohydrate that hangs off of a protein on the membrane of the one type from the A, and from the other type, B. You can actually see both of those. But in incomplete dominance, it's actually more of a blending. It's an in-between thing. For instance, in snapdragons, one allele, big R, encodes an enzyme that makes red pigment, and the other allele, little r, makes no pigment, so it would be white. But the heterozygous individuals are pink, so it's more of a blending effect. Good thing Mendel didn't use snapdragons, otherwise this would have supported the blending hypothesis that existed at the time. Many other books sometimes use all capital letters to do these types of problems, so that R would be, or the red would be big R and white would be big W, because neither of these is recessive. They're both incompletely dominant. So then the heterozygous condition would be RW and would be pink. So I've seen these problems done different ways. It looks like your book still likes to use the capital and the lowercase, but keep in mind, in these problems of incomplete dominance or codominance, the little r does not mean recessiveness. In this case, when we cross a big R little r with a big R little r, we get a 1 to 2 to 1 ratio. Another pattern that we see is called epistasis. Some traits are affected by multiple gene products, an effect called polygenic inheritance or epistasis. So meaning many genes control a trait. For instance, this happens in Labrador retrievers. dogs. So let's say we have a dominant allele, big B, is black fur, and the recessive counterpart is little b for brown fur. So black fur dominant over brown fur. A dominant allele of a different gene, we'll call that gene E, causes color to be deposited in the fur. And the recessive partner, little e, reduces the color. So a dog with a big E and a big B allele has black fur, because those are dominant. Remember, when you're dominant, you only need one copy of that allele to express the trait. A dog with a big E and a homozygous for little b is actually brown, because not as much pigment is laid down. Oh, excuse me. A dog with the big E is, and homozygous for little b is brown. I take that back. The brown is still laid down because the big allele exists. So going to the one I was thinking of here, I got these mixed up. Despite whether you have big B or little b, black or brown alleles, when you get the little e allele, you don't put the pigment down. So you have the yellow fur. There we go. Now I've got it right. So that's kind of an interesting example of epistasis in dogs. A pleiotropic gene influences multiple traits. So one gene affecting multiple phenotypes. Mutations in pleiotropic genes are associated with complex genetic disorders like sickle cell anemia, cystic fibrosis, and Marfan syndrome. Again, one gene affects multiple traits or multiple phenotypes. In Marfan syndrome, mutations affect elasticity of tissues of the heart, skin, blood vessels, tendons, and other body parts. So one gene, many traits here. Some people claim that Abraham Lincoln may have had Marfan syndrome. It's that tall, lanky look. But I don't know that we'll ever know that. Phenotype often results from complex interactions among gene products and the environment, and many traits show a continuous range of variation. So this means sometimes the environment can affect your traits. For instance, hydrangeas, I believe it's hydrangea flowers, their color, despite what gene they have, is also affected by how acidic the soil is. So their color can actually range from pink to blue, depending on acidity of the soil. So there's an example of environment affecting a trait. Some traits appear in different forms, 2 or 3, sometimes more, in a range of differences. This is called continuous variation. The more genes and environmental factors that influence a trait, the more continuous its variation. So in a population, a range of small differences in a shared trait is continuous variation. And generally a graph of this would show a bell curve. Here's some examples. Seasonal change in animal fur color. Spines grow in the presence of predators. And different plant heights when grown at different altitudes. In the summer, the snowshoe hare's fur is brown. In the winter, it's white. In Daphnia, those are little crustaceans that they can develop a tail spine and a pointy head in response to chemicals emitted by predatory insects. That's interesting. And the yarrow plant grew to different heights at 3 different elevations. So remember, not all traits appear in Mendelian inheritance patterns. An allele may be partly dominant over a non-identical partner or codominant with it. Remember, codominant, both alleles are expressed, but with incomplete dominance, it's more of a blending effect. Multiple genes can influence a trait. Some genes influence many traits. And the environment also has an influence on gene expression. So, back to the Menison mucus, revisited there, the mutation allele for cystic fibrosis in homozygotes may persist because it offers heterozygotes individuals a survival against certain deadly infectious diseases, actually. It's called heterozygote advantage when this happens. And people who carry it could have this decreased susceptibility to things like typhoid fever and other bacterial diseases that begin in the intestinal tract. So a Punnett square for someone who has cystic fibrosis generally is when two heterozygotes get together. So we'll say big C, little c, getting together with another big C, little c, and 1/4 of their offspring, 1/4 of the time, will be little c, and they'll have cystic fibrosis. I wanted to take a second to show you a Punnett square here. This is a monohybrid cross here with just the 4 squares. And this was the example of crossing the F1 generation back to our original experiment, big P little p times big P little p. Generally you put one of the partners up here, the male. It doesn't have to be, but generally the male's gametes go up here. So males can either give a big P or a little p. They have sperm that have a big P or a little p. And females have eggs that have either big P or little p as well. So then we fill in our boxes. Big P here. Little p, big P, bring that over. Always write the dominant one first. Big P, little p here, and 2 little p's there. So then we can see that our heterozygote parents produced, with a phenotypic ratio 3:1, purple to white. Or you can look at the genotypic ratio, that 1/4 of them are big P, 1/4 little p, and 2 out of the 4 boxes, or 1/2, are big P, little p, or heterozygous.